So welcome to the second day or the second session of our journal club meetings, which we have scheduled to happen like every Monday evenings. Now, today we are going to have two presentations. The first one is going to be from Dr. Madhu Priyadashini, who's doing her MD at SDM College, Ujire. She's doing her MD on nutrition. So she has taken a unique topic, that is the effect of pomegranate juice on diabetes patients, and to see the effects. When mostly what we tell to patients is, as far as diabetes is concerned, do not take juices. And she wanted to present this particular paper because she found it interesting on the benefits of pomegranate juice consumption in diabetes patient. That would be the first presentation. And the second presentation, I'm going to do the second presentation, which is going to be on chromotherapy. Chromotherapy, yes, very colorful, very interesting, but is it scientific? If yes, how much scientific it is? That's what we are going to explore today with the available research evidences. Over to you, Dr. Madhu Priyadashini. You can start your presentation. Thank you.
Good evening, sir. A very warm welcome to everyone present here in today's journal club. I thank Dr. Venugopal sir for giving me this opportunity to present this article here. Am I audible, sir?
Yes, loud and clear. Yeah, go ahead.
So today's paper is, the title of today's journal club is to see the effect of pomegranate juice consumption on oxidative stress in patients with type 2 diabetes mellitus, a single blind randomized controlled trial. This paper was published in September 2016 in the Journal of International Journal of Food Sciences and Nutrition. So I have attached here the abstract of the paper. So let me go ahead with the introduction. We know that diabetes mellitus, type 2 diabetes mellitus has become a huge concern in recent times. And hello. Yeah, I'll continue. So type 2 diabetes mellitus has been a huge
Madhu, I mean, you've been muted as well. Could you kindly unmute, please?
Am I audible, sir?
Yes, you're audible.
Okay, so we know that diabetes mellitus is a major health problem worldwide. And in 2011, there was 366 million people affected with diabetes mellitus. And this will increase up to 552 million in 2030. So the clinical complications, that is microvascular complications and macrovascular complications, was affected in about 30% to 50% of worldwide population. So the most important cause of micro and macrovascular complications was oxidative stress. So what happens when oxidative stress happens is there is enhanced mitochondrial superoxide production in endothelial cells of large and small vessels, which leads to generation of reactive oxygen species and decreased nitric oxide in our body, which leads to reduced potent antioxidant capacity of the human body. So what is oxidative stress? Oxidative stress, it causes oxidation of the LDL, that is the low density lipoprotein, which is also called as the bad cholesterol. It also leads to atherosclerosis and macrophage foam cell formation. So what was looked upon was the polyphenol content, which is the natural antioxidants present in diet. They help in preventing oxidative damages. So pomegranate, which is a very readily available fruit, it had a high concentration of total polyphenol content and it had higher antioxidant capacity than green tea and red wine. So this study was basically conducted in Iran. So the parameters which was assessed to check the oxidative stress were: one, serum oxidized low density lipoprotein, serum antioxidized LDL antibodies, total antioxidant capacity, that is TAC, and PONA, that is aryl esterase activity of peroxanase. So coming to the study profile or the, let me just briefly tell you about the study profile. The totally 250 participants were assessed for eligibility before starting the study. And from 250 participants, totally 62 people were chosen. So there were 112 people who were excluded and 40 people refused to participate and 21 were not meeting the inclusion criteria. I'll be explaining the inclusion and exclusion criteria in the subsequent sections, and 15 participants declined to participate in the study. So they were randomly allocated into two groups. That is, one is PJ group, that is the group which was given the intervention, that is pomegranate juice. The other group was the control group. Each had 31 participants, and from each group, again, one person declined. So totally 30 participants, they completed the study. So coming to the methodology, this study design was a randomized study, single blind clinical trial. So looking into the inclusion criteria, patients diagnosed with type 2 diabetes mellitus were included between the age of 40 to 65 years old. So they were diagnosed of at least 12 months. So the fasting blood glucose considered here was equal to or more than 126 mg per dl. And looking into the exclusion criteria for this study, participants who had the habit of smoking, who are on insulin for blood glucose control, and any female participants who are on estrogen or progesterone supplements, or any antioxidant supplements, or participants who have other chronic disorders were excluded from the study, and also participants who missed 10% of pomegranate juice consumption or who had any side effects after consumption of pomegranate juice were excluded from the study. Coming to the study design, so there were totally 62 subjects recruited. So the sample size was designed to detect 20 unit per ml difference in 6 weeks of aryl esterase activity of peroxanase between the groups. And the sample size was estimated by 95% confidence interval and 80% power. They were randomly allocated by sequentially numbered containers. So looking into the intervention, that is the control group, they were remained untreated for the duration of 6 weeks, whereas the pomegranate juice consumption group took 200 ml of pomegranate juice per day for a period of 6 weeks. So both the group subjects were told to maintain the dietary habits and physical activity and if medications during the study. The randomization was blinded for the participants. And coming to the assessments which was taken during the study, looking into the anthropometry measures, weight was assessed by balance Sika scale by wearing light clothes, removing the shoes, and height was also measured using stadiometer, removing the shoes, and body mass index or the BMI was calculated by weight divided by square of height in meter square. So coming to the dietary intake assessment or the dietary parameters, whatever was assessed during the study, a 3-day dietary recall was taken, which includes 2 weeks days and 1 weekend day. And the dietary intake was assessed by Nutritionist 4 software. The biochemical parameters which was measured during the study was 10 ml of fasting venous blood sample after 12 to 14 hours of overnight fasting was measured. Serum oxidized LDL, serum antioxidized LDL antibodies, total antioxidant capacity, and PONA, that is aryl esterase activity of peroxanase was measured. So let me just give you a small brief of how these parameters was measured. So serum oxidized LDL was measured during LDL oxidized kit, and serum antioxidized LDL antibodies concentration was measured using the immunoELISA antioxidized LDL kit. The serum total antioxidant status was measured using the commercially available kit, and serum PON1 was measured using the phenyl acetate as a substrate was determined spectrophotometrically as described. And coming to the pomegranate juice, so the pomegranates of Persian species, they were picked by hand, they were washed and stored. The fruits were crushed, peeled and squeezed, and the juice was pasteurized, concentrated and stored at minus 18 degree Celsius. So the concentrated pomegranate juice was diluted 1:5 with water in order to obtain a single strength pomegranate juice. So every week the juice was delivered in tetra packs to the participants, and it was done free of charge. So on assessing the chemical constituents of the pomegranate juice, it contained 2125 mg per dl of total polyphenols and 385 nanogram per ml of total flavonoid content and 5.9 gram glucose in 100 ml of pomegranate juice. Looking into the statistical methods, the analysis done by using SPSS software. I'll be showing you the tables in the next subsequent slides. The qualitative variable stage chi-square test was done to compare the quantitative variables between the two groups, and Kolmogorov-Smirnov test was done to assess the normal distribution of the data. Pair t-test was used to compare the pre and post intervention within the groups. Student t-test was done to compare the mean changes within the groups. So adjustment for differences in baseline covariates and changes in variables during the study was measured by analysis of covariance. So the differences were considered significant at p-value less than 0.05. Coming to the results, the fasting blood glucose mean concentration in pomegranate juice group was significantly lower than the control group. The mean changes of fasting blood glucose between the two groups wasn't statistically different between the two groups at the end of the study. At baseline, the aryl esterase activity was significantly lower in pomegranate juice treated group, and also at the end of the study, the oxidized LDL and anti-oxidized LDL antibodies significantly decreased. The total antioxidant capacity and the aryl esterase activity of peroxinase increased significantly in the pomegranate juice group. These are the tabulations which shows the baseline characteristics of the study population. Coming to the gender distribution, equally male and female participants were taken in the study, like 15 male and 15 female from each group was they completed the study. And also the dietary intake of the study population was tabulated on viewing the vitamin E intake in the control group. It was reduced post intervention. And also tabulation, the third tabulation showed the fasting plasma glucose and the oxidative stress parameters in the study population. So totally, apart from the fasting plasma glucose, the other parameters are, as mentioned, the oxidized LDL, antioxidized LDL antibodies, total antioxidant capacity, and aryl esterase peroxinase activity. These were mentioned in mean plus or minus standard deviation. And coming into the discussion, so there was no adverse effects seen on blood glucose level daily consumption of 200 ml of pomegranate juice, but there was improved oxidative stress parameters significantly. So looking into the main antioxidants in the pomegranates, So the tannins, anthocyanins, and ellagic acid, they contribute to the total antioxidant capacity of pomegranate juice. The anthocyanins absorbed in the glycated forms led to elevation of the glycosides, and the flavonoid metabolites, they have different biological and antioxidant properties than their parent compounds. The most abundant elagitannins in pomegranate juice was the punica lignin, which was responsible for the high antioxidant capacity of pomegranate. And the mechanism of pomegranate juice, like how it improved the oxidative stress parameters, they they showed that it was due to the activity of its high polyphenol content and also its anthocyanin content. There was also previous studies which told that 50 ml per day of pomegranate juice consumption for a period of 2 weeks reduced the lipid peroxidation by 6%. It also increased the total antioxidant capacity by 9% and the activity of aryl esterase activity of peroxidase by 18%. There was one more study which told that 50 ml of pomegranate juice consumption per day for a period of 1 year in asymptomatic severe carotid artery stenosis patients, it reduced the carotid intima media thickness significantly, and also the LDL susceptibility to oxidation and antioxidized LDL antibodies by 30%, 59%, and 19%. It also showed that there was increased aryl esterase activity of peroxidase and improved total antioxidant status by 83% and 130%. There was one more study which said that pomegranate juice consumption per day for a period of 3 months on diabetics did not worsen the parameters, but it reduced the serum lipid peroxides, thiobarbituric acid reaction by 56% and 28%. And there was also increased PONI, that is the aryl esterase peroxidase activity by 24%. We know that type 2 diabetics, they are highly prone to oxidative stress, which leads to glucose auto-oxidation. And also there is increased NADH production and superoxide radicals. And also there is also increased advanced glycation end products and reactive oxygen species. So what is this PONI? We have been mentioning about this aryl esterase peroxidase activity. So this is actually HDL associated lactonase or esterase. It has the ability to hydrolyze the oxidized lipids, and also it prevents HDL and LDL oxidation. So I'd also like to conclude in the study that pomegranate juice consumption for a period of 6 weeks, it had positive effects on serum oxidized LDL and antioxidized LDL antibodies, as well as serum total antioxidant capacity and aryl esterase activity in patients with diabetes. So mainly, we'll have to see two things when it comes to a research paper. One, what is the strength of the study? And the second one is what is the limitations of the study? So the limitations which I would like to see from this paper or from the study is they did not check for HbA1c levels. So that wasn't measured at the baseline and also after the treatment. And there was no suitable placebo drink which was prepared for the study. These were two limitations which I could see in this study. And mainly if we see the strength of the study, very important strength is there was a high percentage of patients who completed the study, and a detailed data collection through face-to-face meetings happened, and having the control group also. So the participants or the study design, which the randomization which was done were the stratified block randomization design. These were the most important strengths of the study. And forgot to mention, the ethical clearance for this study was taken from the Iranian University of Health Sciences, and also consent form was given to all the study subjects, both the control group and the study group. They are also detailedly explained regarding the goal of the study, and then they were proceeded for the intervention. Thank you.
Thank you. Thank you so much, Dr. Madhu Priyadashni. I know it's a bit overwhelming to start with the proceeding and start with the presentation first up. You did really well. Thank you. Thank you so much. And here, one most important thing that's been proved in this study is that we have so many misconceptions, not just about diabetes, but also for other conditions, whether this will work, whether this will not work, whether this can be given, not given. And this is one such study which talks about like pomegranate juice is actually good. It's not only measuring the blood glucose level, as you can see over here. The important etiopathogenesis in diabetes, it's about the inflammation and the free radical damage, and this is actually addressed in that. And one more important thing when it comes to journal club, the most important part is you are not just presenting whatever is being told in the paper. You have to give your own inputs. You have to tell what are the plus and minus, positives and negatives of what exactly the paper is, the paper's positive and negative facts from your perspective. It need not necessarily be the 100% true factor as such, but from your perspective. And Dr. Madhu did a really wonderful job telling about that as well. Now here, there were a lot of statistical terms which was mentioned by Dr. Madhu in the statistics part. I know it's a bit overwhelming, especially if your students, I see like many students are here as well, along with, many doctors and even principals of a few institutions. I'm able to see him. Thank you. Thank you so much for coming. It's a real privilege. Now, for students, please don't get overwhelmed with the statistics part. As I promised in the last week, we will just explore one by one so that in a few weeks you become experts in what these statistical methods are, right? To just name one thing, she was mentioning about the block randomization as a strength of the study. Okay, what is block randomization? How we generally randomize the samples is that, like if we have 60 patients, say for example, if you have 60 or 80 patients, you put them under the randomized computer generator, what happens is we will not exactly get 30 each in each group. It should not be like 30 each in each group or 40 in each group. But if you're block randomizing, like the first 10 people, if they are coming, in that 10 people, 5 of them will be in the control group, 5 of them will be the intervention group. So that is called as block randomization. You take 10 patients as one block, randomize them. You take the other 10, put them in one block and then randomize them so that we get almost equal number of patients in both the group. So that is what is called as block randomization. That's one of the randomization technique. It is not a research, research design or anything. It's only a randomization technique. Fine. Right. Okay. Thank you. Thank you so much. So with that, we'll just move to the second presentation of the day. Right. Second.
Sir, can I ask a question, sir?
Yes, a quick question. This will just take only a couple of minutes.
Yeah, sir. Just a simple thing.
Mhm.
Sir, this is about a type 2 diabetes mellitus, right? And madam has mentioned that it has the insulin taking people, that criteria has been excluded, right? So can I just know a glimpse of what will happen if it given to type 1 diabetes, which means that the people who are taking insulin?
Okay. You want Dr. Madhu to answer, or do you want me to answer?
Sir, anyone is fine.
Okay,
sir.
fine. Now the thing is, as far as diabetes is concerned, now here what we are looking at is the free radical damage. It's not even about what do you call it? It's not even about the blood glucose levels and other stuff. It's about the free radical damage, which is high in type 1 diabetes as well. And coming to free radical damage, now how this happens is whenever there is glucose in your body, whenever there is glucose in your blood or in your system or whatever it is, what happens? That glucose has to get converted into energy, right? So in
Yes.
the process of conversion of glucose into energy, we have this free radicals getting like thrown out or eliminated into the system. Most probably it happens at the mitochondrial level, right? So
Okay.
until and unless these free radicals are overcome, there will be beta cell damage. If it's happening within the beta cells, it's going to damage the beta cells. If it's going to be happening in the muscle cells, it is going to damage the muscle cell where these free radicals are actually accumulating. When it comes to type 1 diabetes, the complication, the risk of complication is much, much higher in type 1 diabetes when compared to type 2 diabetes. So preferably this would be, I mean, even more beneficial for type 1 diabetes. And I don't think like insulin has got anything to do with the, what to say, spike in glucose level or lesser glucose level for that matter. Okay.
Yes.
It is just to keep the group less riskier. It's just to keep the group less risky, because this is something we are doing. It's a out of the box thinking. We are going to give juice to a diabetes patient, which is against the conventional wisdom, the wisdom which has been followed for many years. We are trying to break that and try to find some benefit on how this free radical, how this juice is going to be beneficial in reducing the oxidative stress. For that, we have to be a bit more careful. It's better to be safer than sorry, right? So they wanted to put it in a very safer environment, and they do not want to get into like more complicated patients with more complications or higher drug or medication score and other things. That's one main thing.
Okay.
All right. Hope that answered your question.
Thank you, sir. Thank you, sir.
Thank you. Thank you so much. All right. Now that takes us to the second presentation, and interestingly, that's going to be on oxidative stress as well. Okay, oxidative stress from the chromotherapy aspect. Right. Let me start the presentation. All right. Now, this study is, as I told, it's on the effect of different wavelengths on superoxide dismutase. Now, I just want to take this like some part of it, or at least half of it. It's going to be purely from a journal club presentation point of view. That is, I'm going to tell you the what is the methodology, what is the materials and methods, introduction, and other things. And the final part of it, when we are going to talk about the possible mechanisms and whether chromotherapy is true or not and other things, we are going to go into like more of what to say, a simpler term and discussion. It's not going to follow the concrete research kind of thing. Fine. That's what we did with the hydrotherapy mechanisms last week, right? Just like what we did for the hydrotherapy mechanisms last week, we are going to look at the chromotherapy mechanism this week. This week. Yeah. So here the paper title, Effect of Different Wavelengths on Superoxide Dismutase. Kindly make a note, they did not say the effect of different colors on superoxide dismutase, right? So they are talking about different wavelengths. They are seeing colors as wavelengths, right? Wavelengths. It's not something which is going to increase our, I'm looking at something pleasant, I'm looking at something unpleasant. It's not about that. All colors are wavelengths. All colors are electromagnetic waves, right? And the authors of this paper are Assimi. Now, Assimi and chromotherapy, you cannot separate both of them, right? Because plenty of work is done on chromotherapy, phototherapy, light therapy, whatever you call it, but Assimi would be there from Pakistan University, and has done some beautiful works across 2 decades now, right? So the journal is Journal of Acupuncture and Meridian Studies, and this is PubMed indexed as well, just like what Dr. Madhu had presented. Publishers are from Science Direct, and DOI link I've given. If I join the group Research Alchemist, I'll be sharing this paper as well, right? Yes. Now, I've just put the result as well here, right? So this result show like red color red color found to be having better influence or impact on superoxide dismutase, right? All other colors also had an impact, but not as much as what red color had on this particular enzyme. Fine. So going as per the traditional journal club way, we are talking about the abstract, which is a structured abstract. As I was mentioning last week, abstracts are of two types, structured and unstructured. Structured means it will be having one, like just like a human body, we're having like head, trunk, limbs, and other things, right? Likewise, the abstract would be having the introduction part, methodology, results, and finally conclusion. So that's what we call a structured abstract. If it is unstructured abstract, everything would be written in a paragraph, in just one paragraph without like concrete or different structures on it. So it has got introduction, methods, results, and conclusion. Coming into the main body of the content, introduction. Introduction didn't say anything about chromotherapy, right? Introduction says about superoxide dismutase, which is an antioxidant. What it actually does, it converts superoxide to oxygen and hydrogen peroxide. And hydrogen peroxide, again, like within the cell, we have this catalase, which is going to break hydrogen peroxide into hydrogen and water. This is like what you can say, continuation of what Dr. Madhu Priyadarshini was taking short while ago. So we are drinking a juice, we are drinking some fruits, vegetables, or whatever it is, which has got the antioxidant capacity. And once we are taking that, that is going to create a few changes at the cellular level. So that is who's taking care of that? These enzymes. Superoxide dismutase catalase, these are the enzymes which are taking care of these reactions. And the authors mentioned the objective or the aim of the study is to see the SOD activity under different wavelengths of light. Okay, now materials and methods. Okay, now this is what is slightly interesting part. Now this is a lab-based study. Now this is not done on, it's not a human trial or anything of that sort. Just like how medicines before coming into the market, it is, I mean, it run through these cell lines, right? Cell lines, animal models, and only then it comes to patients. Likewise, chromotherapy, color, electromagnetic field, electromagnetic radiations, all these things can be done at the lab setting itself. Now, how they have done this is, they mix 1 milligram of superoxide dismutase with 10 ml of distilled water, and they put it in 7 different test tubes, right? 7 different test tubes of 2.5 ml each. It's not like from 10 ml they took. This is like kind of a proportion we are talking about. It's not like 2.5 into 7 or something else. Don't go into that. If they've taken 10 milligram, they would have mixed it with 100 ml of distilled water. Not a problem. So we have 7 different test tubes. One is for the control. One doesn't have any wrappers around it. And the 5 test tubes, they're wrapped it with different color cellophane tapes, like cellophane filter sheets, like what we used to wrap with the glass paper, right? The different color glass papers. So the 5 colors are blue, green, purple, yellow, and red. So from the visible spectrum, they've started from like from the blue end all the way till red end, and in between as well, they've taken everything along with one control group. And they expose, right? They expose all the test tubes for 10 minutes under 12-watt full spectrum incandescent light bulb. Now, why I'm stressing on this full spectrum incandescent light bulb is because I tried doing a study, like similar study I wanted to do. I wanted to test whether green color, we say like green color has got antibiotic property, right? And most of you would be knowing, I mean, I was working in a molecular bioscience lab at SVSI University, right? So that's where I learned ABCD of research. And then our HOD was preparing cell culture, bacterial cell culture. So he was preparing the culture. It's a kind of a tedious process. It takes like 2 to 3 days where we just grow the culture and put it in, put food for the bacteria and then put it like full beaker full of bacteria he had. I was looking at the beaker and I asked like, sir, I have one doubt. He asked, what doubt? I told like, if I put a 1 drop of Dettol into that, what will happen to all the bacteria? Because what the advertisement tells us is Dettol kills 99.9% bacteria. So obviously I had the curiosity, like if I'm putting Dettol into that, what will happen? He looked at me and instead he told like, you will kill all the bacteria if you put Dettol into it. Then sir, I have my second doubt. I told like, in chromotherapy, they taught me that green color helps in, green color has got this antibiotic property or the antiseptic property, it kills bacteria. What if I just wrap a glass paper, green glass paper around it, and I just put it in sunlight and whatever it is, green color charged water. If I'm putting a green color charged water, will all the bacteria die? He told, I don't know, you try, right? I didn't know that this happened somewhere around 2015 to 16. I didn't know like chromotherapy, there were studies before, and Asimi has done everything, all of these before. I had no clue. I wanted to try that. Now I did everything perfectly except one thing. Where I missed is this full spectrum incandescent light bulb, right? What I did, I wrapped like 3 to 4 different beakers. One is for the control group, one is at the blue, one is at the red, and something in between. And all these things, the culturing, everything was done. Now, what my HOD told us, you cannot keep it in the sunlight because it has to be continuously, you have to churn it, right? You have to continuously churn it, allow like 12 hours for the whole thing to happen. It has to happen only in the incubator under certain degrees. So I wanted to do it indoor. Now, what I did, I put it inside, inside the incubator, and I connected a light bulb. Okay, all the beakers are wrapped around with red color, blue color, green color, and normal glass paper, right? Everything was inside, and then I put a light bulb. I put the costliest light bulb inside the incubator. Okay, now the problem is that next day morning I came, I just compared all the samples. Every sample, the growth was the same. The color had absolutely no impact on it. Then I realized the costliest bulb what we have is a monochromatic light bulb. Whatever the LED lights we use, either it be the bulb or the tube light, it's LED light. LED monochromatic light has got only one color. It will not have the whole spectrum. Okay. Previous day we used to have this round bulb, right? The round bulb with 80 watts and 100 watts bulb. That has got all 7 colors of the spectrum. But the monochromatic lights will not have all 7 colors. So, what to say? If you put a prism and white light passes through that, it has to give like 7 different colors. But if you are putting this monochromatic light, it will not give 7 different colors. To put it in the simplest way, right? The 7 colors will come out of a light spectrum, and you are covering it with a say like red color beaker covered with red glass paper. What this red glass paper does is it absorbs only red color. It will not absorb any other color. That's how it works. Why I'm telling you this is, if you want to do this, if you have a proper biochemistry and microbiology lab at your university, at your college, you can try this. But please make sure you do not use a monochromatic light bulb. You have to use a full spectrum incandescent light bulb. Okay, a full spectrum incandescent light bulb you have to use if you are going to do a study on chromotherapy. If it's indoor, if you can do it in outdoor, it's much more easier and better. And here they did for only 10 minutes. Now my study was on bacteria culture. So if you're doing on a bacteria culture, it has to grow for at least 24 hours. So that is why I had to put it in the incubator and all these stuff. But here they did for only 10 minutes. And whatever the result came out, they measured with a spectrophotometer. And what the results came? So this was the result, as I told you. This is the control group. Everything had an impact, but red had the most impact. Now talking about when compared to yellow, this is twice as much. This what to say? All these pillars might not do any justice as such, but the amount of absorbance of the results is 2 times as much as this yellow is. Okay, So the superoxide disseminate, this antioxidant responded the most with red color wavelength, implying that free radical elimination may be easier. Yeah, sorry, typo, easy. It may become easier and rapid with an application of red color in the body. And that might be the reason why pomegranate juice has found as similar results or similar effect. Anyway, I don't want to compare this and that, but again, that's what we learn in chromotherapy, right? So the color get passed on to the cellular level. Fine. Now this is one thing. Now what we have found here is red color has got antioxidant property. One thing. Okay. And similarly, now before going into the discussion part, is this a discussion? Okay. Yeah, before going into the discussion and the mechanism part, just want to tell about another study. Again, Asimi et al., Asimi and associates have done another study. So what they found was two colors. Sorry, what happened here? So two colors, I mean more than saying two colors, we have an where we all right. Now this study was done with two crucial things. One was here we taken an enzyme superoxide dismutase. That's one part of it. Here they have taken two important things. One is concerned with diabetes patient, the other with obesity, right? So what are the enzymes they have taken into consideration over here is glucose oxidase, okay, glucose oxidase, cholesterol oxidase plus cholesterol esterase and lipase, right? So what they found was dark violet color was found to be beneficial in improving the cholesterol metabolism. To put it in the simplest way, I don't want to go into the enzymes and other things. Dark violet is good for cholesterol metabolism. Likewise, purple color is good for glucose metabolism. Fine. So if you have a diabetes patient, go for purple color thing, purple color charged water and other stuff. And obesity, go for dark violet. Okay. Yes. Now coming into the is it moving? Yes. Okay. I don't want to call it mechanisms. I just want you to understand a few of the terminologies. Terminology. I mean, again, it's going to be about the mechanisms. Right. I'm very much pessimistic about the science behind chromotherapy. Right. That is where the actual search would come. If you start believing in something completely 100% without questioning it, then that is not the true knowledge. That's not the real knowledge. You are just you're accepting it just for the sake of accepting. That is where my yoga research journey started as well. I was pessimistic about yoga. I thought like, why there is so much of hype about yoga? Yoga is like something similar to physical exercise, and people keep saying yoga does this, yoga does that, and everything. That is when my journey into yoga research started. I went into the yoga anatomy, yoga physiology, and into the mechanistic part, and every single aspect of yoga. Only when I explored, only when I uncovered, I got the confidence to say that yoga is the best. Likewise, chromotherapy. Many of you, when chromotherapy class is happening, will be thinking like, what story is going on? This story, that story. How can this create that impact and all these things? Even I had all those thoughts, and that is why I wanted to dive deep into chromotherapy. I wanted to do a research to see, to show that it actually works, right? Now, there are a few concepts we have to understand. Now, why are we sitting in a chromotherapy class and laughing within ourselves at what story is going on? Because we believe in the Newtonian concept. Now, what is the Newtonian concept? We all know Isaac Newton for one thing. What? Apple fell on his head. And the second thing is about Gravity, right? So gravity, what are the objects which can fall under gravity? Anything with mass, right? Either it be the solid, liquid, or whatever, anything which has certain amount of weight, a physical object, only that can get acted upon the gravitational force. That is what the conventional medicine or the modern medicine accepts in the Newtonian concept. I will believe in only what I see. I will not believe what I don't see. So they don't believe in the energy concept or energy aspect. Now, there's a second scientist who came into the picture who's going to, what to say, who changed a lot of things. That's Einstein. Okay. Einstein. The moment we say Einstein, the first thing which flashes is light bulb. The second thing which flashes is E is equal to mc square. Watch a documentary on how this formula came out. Like if you have time, I think it's a Discovery or National Geographic Channel document. It's such a beautiful documentary. For us, it's just 3 letters. E is equal to mc square. What's the big deal? But why he spoke about energy? Why mass is there? Why not some other thing? And what does that C has got to do there? Why we took the speed of light there for calculating energy? Everything was there in the documentary. Now, anyway, all these things I've taken from a research paper. I'm going to share that research paper with you for sure. Okay. Now, what Einstein told was, now in the same formula, we have this M, we have this mass there, right? What he told was, if you are keeping an object in an electromagnetic field, at the speed of light, right? At the speed of light, what happens is the vibration is going to increase. Okay, the oscillation or the frequency, the vibration is going to increase, and that is going to give energy out. And he went like one step ahead, he went one step forward. And what he told was, if you can vibrate at the speed of light, the mc square, c square is nothing but the speed of light. If an object with a certain n number of n gram or n kilogram of mass, if it can vibrate at the speed of light, at the square of the speed of light, that loses its physical object and turns into energy. Okay, so that is what we are reading in yoga. What happens at the state of samadhi? Right? What happens at the stage of samadhi when people leave out their physical body and then go into the subtle body, like I've heard about like Aurobindo, Ramana Maharishi, and other people, right? So what we have seen is they have lost this physical body and turn into energy body because they know how to alter the frequency of the body. They know how to alter the frequency of the mind. Okay, fine. That's Einstein approach. I'm very sure the moment you can do that, what Einstein says is you can time travel. Fine, you can time travel. Einstein, I mean, as far as I know, is the only person in the scientific history, the scientific community, a scientist telling about time travel. It would be Einstein, and he believes in this energy, the concept of energy. If a body can vibrate at the speed of light, it can time travel because it gets converted into energy. It might be a bit too much for us because we're still in the Newtonian concept. And there's kind of a link between Einstein and Newton concept. There's something called Fraunhofer lines. What this Fraunhofer lines says is, if you are burning a physical object, say like take a metal, take any metal, when you are burning it, what happens is each metal will be burning in a different colored flame, right? Different colored flame, which is represented using the Fraunhofer line. So what it says is, I mean, the linking concept here is there is a mass, there's a metal, you're burning it. When you're heating something, what happens? The frequency increases, isn't it? When you're boiling cold water and making it a hot water, roll boiling it, what happens? The sound, whatever you're hearing, it's about the electrons getting excited. The frequency within changes. Likewise, when you heat up the metal, the frequency changes, the frequency increases, and that turns out emits different colors of the visible spectrum, where we are moving from the Newtonian to the Einstein approach. So every physical body has got set frequency. Likewise, Every physical body, in the sense like every physical organ, has got a set frequency. It has got that set frequency. If that frequency is getting disturbed, we can replace it with another frequency, or whatever frequency is lost over here, we can replace it with that. Okay, fine. So that's about the Newtonian concept of Einstein. Oh my God, electrolysis. Fine. I'll just quickly finish this electrolysis in 2 minutes. You're having a saline solution, like imagine a beaker, there's a saline solution, and you pass electric current into it. What would happen? Nothing would happen. Current would pass. If you touch, you will get shocked and electrocuted and die. What if you put a cathode and anode into that solution and then pass electric current? What happens? Cations. Now, this is high school physics. I mean, yeah, physics or chemistry, whatever. I think it's high school physics. Very, very basics I'm talking about. This is not rocket science. A very, very, very basic thing what we are missing to see, right? So now if you put a cathode and an anode and then pass electric current into it, cations move towards cathode, anions move towards anode. So even when you're passing plain electric current, there will be movement. The only thing is we will not be able to see that. We will not be able to measure that. We will not be able to quantify that. Right? So that quantification happens the moment you insert the cathode and anode into it. As simple as that. Likewise, when you want to measure a few things, we need such specific instruments to quantify it, to measure it, and to scientifically prove it. And I think that's what happened in these particular studies, right? So what happened in these studies is when you pass an electromagnetic color, electromagnetic radiation, which is in the form of the color, if it is passed through a solution, it causes ripples in it. It causes changes in it. And if it causes changes in an enzyme, what happens? The enzymatic reactions would start to happen. The enzyme would start to get activated. That is the reason when you pass red color, your enzymes for antioxidant enzymes, it's going to get activated. You pass a dark violet color, enzyme for cholesterol metabolism, it's going to increase. Likewise, when you're passing purple color for glucose metabolism, it's going to get increased. Okay, so this is all because electric current is electric current is passing through our body. Not exactly electric current, electromagnetic current is passing through our body. So if it is passing through our body, naturally what happens? It's going to cause all the electrolytic changes within the solution. What solution we are talking about? The body is made up of water, right? Yeah, I mean, we'll talk about that a little bit down. Body is made up of water. So obviously, when an electric current is passing through us, we are going to get shocked. We are going to get electrocuted in a good way. And the fourth thing is about the photobiomodulation. More often than not, we are talking about the light mitochondria interaction here. Photobiomodulation, classic example would be photosynthesis. What plants do? They use sunlight to prepare, to make it and convert it into chemical energy called food for it. Okay, food for it. That is what we call as photobiomodulation. The light hits on the photoreceptors in the cell at the molecular level, whatever it is, it's going to hit the photoreceptors and cause a certain biological changes. The biological changes could be the DNA, RNA synthesis. Now, DNA, RNA synthesis, we all know that light radiations are cancer inducing. We say that for X-rays, right? We say that for X-rays, we say that for microwave rays, for all rays, we accept that it is going to make cellular changes. It's going to make DNA changes. But when it comes to visible light, we don't agree and accept to it. Please do accept and agree. It definitely has an impact on a DNA, RNA synthesis, cellular proliferation, and at the molecular level, the cyclic AMP kinase pathways as well. Okay, so it's because the light interacts with the mitochondria. I'm sorry. Okay, so 5 more minutes. I'll just quickly finish. And the other thing is about the neurohormonal pathways, suprachiasmatic nucleus, right, which senses light, whether it is day or light, your circadian rhythm, the chronobiology, everything is dependent on your SAN in your hypothalamus, suprachiasmatic nucleus in your hypothalamus. Through the serotonin and melatonin pathways, it's going to create the neurohormonal changes. And this is mostly helpful in the psychological aspects, right? And hydrochromotherapy. Now, what Dr. Asimi says is the moment we learned and understood that water can be charged through chromotherapy through color, it's a quantum leap in treatments. Okay, it's actually a quantum leap in treatments, because the moment we are going to charge the water, either it be in the bottle or in our body, what we call as intracellular structured water or exclusion zone water, they're calling it as an energy reservoir inside every cell. Now, this energy reservoir, this energy in the reservoir increases its potential energy whenever a specific light, specific to that particular organ, to that particular property, hits upon it. Okay, so these are few of the terminologies or the mechanisms which we can simply put it. Like there could be like hundreds and hundreds, and this is all I can go through, I can find, and I was convinced. And these things are quite sufficient enough to show that physical body is more than just physical body. It is an energy body as well. If we can understand the vibration and the frequencies, and if there is adequate amount of frequency push into it, which can be active, which activates a set of things. We are talking about the electrolysis or the photobiomodulation, neurohormonal pathways, or the hydrochromotherapy part. All these are different ways through which it's going to be beneficial for chromotherapy as a management, right? Okay, now fine. So the discussion part, yeah. And then benefits, the common benefits. This again, I'm not restricting it with the only the paper which I had mentioned. The common benefits are the bacterial culture. As I told, green color, I think Asimi has told like 15 years back, green color, it's going to reduce the bacterial growth, right? So green color has got antibiotic or the antiseptic property that has been shown. Wound healing, red color. Now I will share the paper. I will share the paper with you. Wound healing, red color. Now red color, immediately what we would say is, oh my god, red color for wound. What happens if the blood vessel gets dilated? All the blood loss would be happening through that wound area. It's not actually that. That there is like nitric oxide synthase. When we saw the hydrotherapy for hot application, what I told was, whenever keeping a hot application, what happens? Your skin goes red. How it is happening? Because of nitric oxide, because of the nitric oxide synthase. Now, nitric oxide synthase, there are three types. One is the inflammatory or induced nitric oxide synthase, INOS, ENOS, NOS. NOS is the neuronal nitric oxide synthase. ENOS is the endothelial nitric oxide synthase. That is, for wound healing, you need this endothelial nitric oxide synthase. That is, the smaller endothelial blood vessels, they have to get dilated for the wound healing to actually happen. Okay, so for that, we need red color. Now, this ENOS with the accelerated electron transport, anti-inflammatory property, and as we just now saw, the antioxidant property, that's going to be beneficial in wound healing. And for cancer cells as well, they are found with the breast cancer cell lines and also the epidermal melanocyte cell lines. Enzymatic reaction, as we already saw, there are plenty of enzymatic changes which are very much possible through colors, through chromotherapy. Now, again, if your institution has got this facility to do what to say at a laboratory setting or for a human trial as such, it's very well worth doing it. A simple study, a very simple study, you can finish the study in 2 to 3 days and you can send it for publication. Okay, as simple and straightforward. And psychology, obviously, as we know, for fear, anxiety, PTSD, and other conditions, this is very much, chromotherapy is very much beneficial. Okay. So yes, so that's basically it. Thank you. Thank you so much. Thank you so much for your patience. And yes, as usual, so we have finished bang on time at 9 o'clock. If you have any questions, we can take questions for a couple of minutes and then we can wind up. Thank you. And if you have not already joined the Research Alchemist group link for all the students who just want to join there, thank you, thank you, thank you so much. I'll be sharing the papers in that group. Okay.